Hi, class. Let me just get this started. Hello, class. So this is Friday's lecture, and we're going to just pick up where we left off. The where we left off was talking about how proteins synthesized on the ER get into the ER, or in some cases, partially into the ER. So we talked about ER lumen proteins, that is, proteins that go into the core of the ER, the lumen, and ER single pass transmembrane proteins. And so these are a type of transmembrane protein, the simplest type, I guess you would say, where there's just ER part of the protein and a cytosolic part of the protein, um, and a transmembrane domain. And we talked about uses the same enzymes for both, but what's different is that for a single pass transmembrane protein, in addition to having a signal peptide, which is a type of start transfer sequence, you also have a stop transfer sequence shown here in orange. And so the start transfer sequence does the same thing for both types of proteins. But if you have a stop transfer protein, then the translocation stops and the uh, single peptide gets cleaved off by a single peptidase, and you're left with this final product. So notice that, you still translate the outside part. It's just that it's no longer being pushed through once the hydrophobic part has interacted with the membrane and the translocator. So you can get more complex topologies, and I won't talk about those, but they're mostly involved with having additional start transfer sequences that are not single peptides. They're not right at the end, but they're after the hydrophobic. So once you stopped the translocation, you can start translocation downstream, and that'll leave a loop outside. And they talk about that in this uh, video, but you're not responsible for that. Let's see. So another big thing that happens for ER proteins, particularly the transmembrane ER proteins that are made, is that they are glycosylated. And we've talked about protein glycosylation a few times. If you remember, that's one of the ways that amino acids on a protein, specific amino acids on specific proteins, get modified. And it's now time to tell you that there's two basic types of glycosylation. Glycosylation that um, uh, happens on the amino acid asparagine. That's called N-linked. And then there's also glycosylation that happens on serine or threonine. That's O-linked. And it's a nitrogen and asparagine that gets the oligosaccharide, and it's an oxygen in serine or threonine that gets the oligosaccharide. But we won't talk about this now. We're just going to focus on this, the N-linked ones. And that's because the N-linked ones initiate all in the ER, and the O-linked ones initiate later in the Golgi. So it's kind of an interesting mechanism how this happens. Um, uh, the oligosaccharide is built on a lipid called dolichol. So this is just a lipid in the membrane. It's got a head group that's pyrophosphate, two phosphates, and then sugars get added to the second phosphate. And you get this branched oligosaccharide, multiple sugars linked together. And you can see here's one branch. Here's another one. So there's an enzyme called oligosaccharyl transferase, or OST enzyme, that just transfers from the phosphate onto the asparagine. And that's how you glycosylate or N-link glycosylate an ER protein. This particular sugar can be modified further. Sugar's added and then subtracted in the Golgi. So it's not always the end of the N-link modification in the ER, but it's the start of it. So all proteins that are N-linked, glycosylated, that initiates the Golgi. So as it says here, proteins and lipid glycan, it generates separately. And then the glycan, which is the oligosaccharide, is transferred onto the asparagine. I think I said that you can't really flip lipids that have sugars attached, but it turns out there's special flipases that do that. So when I said that, that was not really correct, because there are ways to do that. And what reminded me of that is that, in fact, for this oligosaccharide, that happens. It gets started built on the cytosol side. Sugar gets flipped and then more sugar added. That's all before that sugar's transferred. Okay. So another thing that's happening besides glycosylation is folding of proteins. So this is kind of like in mitochondria and not like the peroxisome or the nucleus. But the protein is unfolded. If you think about it, it's not really, doesn't even exist. As it's being made, it's being pushed into the ER. So it's pushed in an unfolded form. And then it's folded by a chaperone, very much like what happens in mitochondria with HSP70, except that the chaperone here is just, it's called BIP, for binding protein, BIP. And what it does is it folds the protein. And how do you fold proteins when there's so many different structures? What does an enzyme do to fold it? Well, the bottom line is it prevents misfolding of the protein is what it's doing. Protein will always fold in some way, but you want it to fold, if you remember, in the most thermodynamically stable form, because that's going to be the functional form. And how do you get it there? Well, you get it there by making sure that the folding happens as slowly as possible. So when it happens slowly, it's going to get to the most stable form. If it happens fast, it's just whatever form happens to be close by. How does it slow the folding? Well, BIP is a protein that is bound to either ADP or ATP. And when it's bound to ATP, it can bind these hydrophobic patches in the protein, and ATP hydrolyzes when that happens, and you get a stable BIP ADP complex. And that prevents that hydrophobic patch from forming hydrophobic interactions with another hydrophobic patch, because it's already associated with BIP. Now, this on its own would not cause it to fold properly. In fact, it would prevent it from folding at all. And that's what it's doing. It's kind of slowing the folding. It's like a timer because eventually that ADP will be replaced by ATP, as is shown here. And you get BIP when the BIP ADP that's bound here converts to BIP ATP after some period of time, then it's released from the hydrophobic patch. And so now that hydrophobic patch has time to find another hydrophobic patch. So what this does is, for example, allow disulfide bonds to form electrostatic interactions to form first. Those are more specific before the hydrophobic patches form. And the hydrophobic patches then form. If it's a weak hydrophobic interaction, it may come apart and BIP may bind to it, giving it time to move around and be closer to where its real partner should be. And once the protein is fully folded and the hydrophobic patches, you remember, are mostly on the inside of a protein, then BIP will no longer bind to it, leaves that protein alone, and it's a fully folded protein. So let's see. Another thing while we're on the subject of chaperones like BIP and how they work is to talk about the unfolded protein response. The unfolded protein response is a way of turning up chaperones under stressful conditions. If you have stressful conditions like high temperature, folding is not really happening very well. And so you have sensors for misfolded proteins that activate in the ER. And when you have a lot of unfolded proteins in the ER, that sends a signal, actually a series of different signals to the cytosol. One thing it does is it inhibits protein synthesis, but the main thing it does is to activate expression through transcription factors of chaperones. So you get more chaperones expressed when you get more stress. And if you think about it, that makes sense. You need chaperones to deal with unfolded proteins. If you have a lot of stress, you start to see unfolded proteins and you make chaperones and enough chaperones to fold them normally. Unfortunately, for example, in type 2 diabetes, if you have so much unfolded protein as you do in type 2 diabetes, if you remember that's insulin resistant, those patients, it's not like they don't make insulin like people with type 1 diabetes. They make insulin, but it's not functional. They tend to make more and more insulin in the pancreatic cells. And all that insulin, a lot of it is then overwhelms the chaperones, so you induce the unfolded protein response. But if eventually there's enough insulin produced, the second trigger of the unfolded protein response seems to be cell death. And actually, for individuals with type 2 diabetes, the eventual death of enough pancreatic cells can make their diabetes insulin dependent because the large amount of UPR is triggering death of the insulin producing pancreatic cells. So we want to move now to a new topic, and that topic is the way proteins move from the ER to these other targets. And that's called the secretory pathway. And while we're at it, we're going to talk about the way proteins move from the plasma membrane or from outside the cell into the cell. And that's called the endocytic pathway. And these are, and if you want to think about it, two different ways that proteins start from one place and end up in other places. As we talked about before, proteins made in the ER can go into the Golgi and from there to a lot of different locations. And it's noted, it should be noticed that, well, it's called the secretory pathway, probably because a lot of the proteins that go through this pathway end up secreted, that is in the extracellular space. But also many of the proteins that go through this pathway don't go all the way to the end. They are, for example, transmembrane plasma protein, transmembrane proteins on the plasma membrane, or proteins in the Golgi, endosome, and lysosome. So here's a very complicated diagram, and here's one that's a little bit simpler. Let's talk about this just to remind you what this pathway is like. All these proteins that are made in the ER, which is shown here on the left, move to the Golgi. So they move really to one place. But from the Golgi, and particularly from what's called the trans part of the Golgi, they move to the endosome. It's not shown here, but they also move to the lysosomes we'll talk about, and they'll move to the plasma membrane. So those are the three things. And some of them that are moving to the plasma membrane are actually secreted into the extracellular space. So you call that process exocytosis. When cells are moved to the extracellular space. And meanwhile, you get this other process with a similar name, endocytosis. But you notice the endo, the cells are coming in to the cell. So not starting at the ER at all, but starting at either the plasma membrane or actually extracellular proteins that are moved into the cell. And they go to the endosome and then to get recycled or to the lysosome to be degraded, as we talked about earlier part of the class. So this leads us to one sort of key question, which is all these proteins are moving to different places through these pathways and this pathway. They are all moving on these vesicles, as you can see. Vesicles from the ER, these little circles, to the Golgi, vesicles between different parts of the compartments of the Golgi, and then vesicles to plasma membrane, and also vesicles moving in from the plasma. So how do these vesicles move from one compartment to a specific destination? How do you make sure they carry the right proteins? That's what we'll talk about here. We'll come back to this picture later. Actually, pretty quickly. So here is the ER. You've seen kind of just a small part of the ER here. And then that part of the ER that is near the nucleus is also connected to this organelle called the Golgi, which is a unique organelle in one way, in that it's not a single compartment. There are multiple compartments, like layers here. And here's a good image where it shows the Golgi is really a stack of these compartments called cisternae. Cisternae. And it almost looks like the Golgi is a stack of pancakes. The proteins move then from the ER first to this bottom layer here that is much less smooth than most of the cisterna, and that's called the cis-Golgi network. What's happening is vesicles are budding off the ER and then merging with each other and larger structures to form this thing called a cis-Golgi network. And then on top of that are more mature and flattened formed. And you can see a lot of vesicles around here as well. So these vesicles are called anterograde transport, the ones that move from the ER to the Golgi. And there's also a lot of vesicles that are moving from the Golgi back to the ER, and that is called retrograde transport. And you can see that that's a complex process, or involves a lot of different vesicle switches from one compartment to another, but always in the direction back toward the ER. And so that's why it's called retrograde, because you think about ER to Golgi as the natural forward or anterograde, and retrograde and reverse we call retrograde. Anterograde, retrograde. Let me just gonna pause the recording, get a little more water. Okay, we are back, and we want to talk more about then the Golgi as a what's it doing and how is it structured. So one of the principal roles of the Golgi is protein modification. As we mentioned, the N-terminal or N-linked glycosylation continues to add sugar to those ones you started in the ER. You get O-linked glycosylation. One reason for all these different compartments seems to be that a lot of the modification is multi-step. So as a protein is moved from one cisterna to the next, it encounters different enzymes that then introduce different modifications. And it's in that programmed order of modifications that it needs to properly be completely modified at the end. So how is this kind of structure formed, and also how do proteins really move? You notice there's just these vesicles shown here. So how do you get proteins from, say, the cis-Golgi network to the next set of pancakes called the cis-Golgi, and then up to the medial Golgi around here, the trans-Golgi, and finally this kind of more twisted cisternae up at the top, the trans-Golgi network, which, as we said, is where vesicles leave to go to the plasma membrane and to the endosome and lysosome. How does the protein move if there's no vesicles shown here? Well, the current model is called the cisternal maturation. And what it says is you have this stack of pancakes, and you're always making a fresh pancake, a new cis-Golgi network, because new vesicles are always leaving and forming into a new cisterna. If you think about it, that new pancake will be put on the bottom of the stack. And when that happens, this model says, the former cis-Golgi network has now become a cis-Golgi. It's moved up in the stack. And you might say, well, that sounds good, but it doesn't have the enzymes that the modification enzymes needed for a cis-Golgi cisterna. We'll just have the ones of the cis-Golgi network. But during the time that the new cis-Golgi network is being made, vesicles by retrograde transport are carrying all those cis-Golgi enzymes into the cis-Golgi network cisterna. And so by the time it moves up to the second spot, it can do the job of a cis-Golgi. And the same thing is happening with each stage of maturation. Medial Golgi is sending enzymes to the cis-Golgi. The trans-Golgi network is sending enzymes to the trans-Golgi. So you're always changing the character of the cisterna, and the proteins that are carried in it from the ER are carried along. It starts when it's a cis-Golgi network and continues until that same cisterna is now a trans-Golgi network. And so it's able to be exposed to this changing variety of modification enzymes. So that's the cisternal modification model. And you can see retrograde transport has a big role in cisternal maturation, and it has some other roles that we'll talk about in a slide or two. But before we get to that, we want to just talk about the mechanism of how these vesicles form and the first kind of level of specificity. There's two levels of specificity. You got to get the right proteins in the vesicle, and you got to get that vesicle to the right place. So we're going to talk about the first one now, and the second one in a few minutes. So to get the vesicle to form, it always starts with a GTP binding protein. We'll call it sometimes GBP. And it's like any of these GTP binding proteins. It's in two forms. And it's associated with GDP in the form where it binds to the membrane, but then it gets GTP substituted in for the GDP and changes conformation, still bound to the membrane. And that's shown here as these little blue ovals. It's really once it's in the GTP form that it binds to this second kind of protein, which is a coat protein. Turns out every donor organelle is making its own coat proteins or with some variant membranes in them. For example, the ones coming out of the ER lumen have the COP2 coat protein, and so they're called COP2 vesicles. And as a result of having COP2, COP2 can bind to short amino acid sequences. They're only there in the proteins you want to move from the ER to the Golgi. It tracks all those proteins. There's a lot of ER proteins that you don't want to move, and they are not going to bind to COP2. In particular, I think I should say, I'm talking about transmembrane proteins for the moment. So a transmembrane protein, it's there in the ER, but you want to move it to the Golgi, will have a short amino acid tag that binds to COP2. And that's shown here. This is a transmembrane protein meant to be binding to COP2. So even while the GTP binding protein is the thing that gets it started, it's really the coat protein that is giving it that specificity for cargo specificity. And these transmembrane proteins that are captured by the vesicle, they're called membrane cargo protein. And you also capture a different kind of transmembrane protein called a membrane cargo receptor protein. And they also have the same short amino acid sequence that targets them to coat protein, but they're not there to travel as much as to carry other proteins. So particularly you want to sometimes carry soluble proteins. Those proteins are not going to be able to bind to the coat protein, which is um, outside the membrane, but they'll bind to these membrane carboreceptor proteins. And of course, again, you got a sequence here that's bound to the membrane carboreceptor, and then the receptor itself recognizes short sequences in the soluble proteins that you want to move. And any soluble proteins you want to stay in the lumen, those wouldn't have that same signal, so they wouldn't bind to the carboreceptor protein. And then the third class that the V-snare protein attracts is called V-snare proteins, which we'll talk about maybe today, maybe Monday. Now, another thing that the coat protein does, it seems like it's got a lot of big jobs on attracting the cargo, but it's also deforming the membrane into a vesicle. So once it starts to bind, multiple proteins are forming. They're forming a mesh, and that mesh is actually deforming the membrane into this sphere and eventually pinches it off, and it's a vesicle. So another important thing to tell you then is the names of the different kinds of vesicles. They're all pretty much named after their coat protein, like HOP2. The retrograde vesicles use the coat protein COP1, similar name, but a different protein. These vesicles that leave the transgolgi and go to the plasma membrane, they have a protein called these are exocytic vesicles, or they're sometimes called secretory vesicles, but I prefer exocytic vesicles. There's an exocytic vesicle coat protein. So each is targeted by a certain coat protein. Now, one sort of exception to that is you get proteins going to the endosome and to the and also proteins coming in by endocytosis. Turns out they use the same coat protein, even though they're gathering different cargo and sometimes going to different places. Sometimes they go to the lysosome. So what happens here is adaptin proteins that interact with the clathrin proteins and give it its specificity, as we'll talk about. But here you have 1, 2, 3, 4, 5 different kinds of vesicles, all carrying different cargo from different donor organelles to different targets. So yep, here's a good time to talk about those adaptins. As we said, it's always the same coat protein clathrin, but in this case, the clathrin is associated with AP2, in this case with AP1 to go to the endosome, AP3 to go to the lysosome. It's also interesting that all the cargo that we've talked about, it's a particular short amino acid code that the membrane cargo receptor binds to. In the case of these AP1 clathrin things, the cargo receptors that they bind to don't actually recognize amino acid as much as a glycosylation event. That is mannose 6-phosphate. So any proteins that get mannose 6-phosphated, if they're in the transgolgi network, they're going to move to the late endosome. And that you don't really get that M6 P in that form until in the trans-Golgi network. So the trans-Golgi, these earlier forms, they have glycosylation, but they don't have M6P at the end of the oligosaccharide. So it has to be sort of the terminal or end. And we'll talk more about what is happening in the endosome lysosome later. But one thing to talk about here is these retrograde vesicles, like these going from the trans-Golgi network back to the trans-Golgi, and also all the other steps in retrograde. I mean, we already talked about their role C in cisternal maturation. You also sometimes get proteins that are missorted. So this is a kind of quality control that moves proteins that get into these vesicles by mistake and end up in the Golgi by mistake. They can be recognized as non-Golgi ER proteins and just moved back by retrograde transport. So that's A. Sometimes you want to move proteins into the Golgi for the Golgi's job of protein modification, such as glycosylation, and then move back through retrograde transport into the ER where they're actually functional. So some proteins have to go into the Golgi just to be fully modified. And finally, you get all these enzymes like are shown in this list that are made in the ER and moved to the Golgi, but they're really mostly functional, to make a vesicle and fill it with cargo at the ER. So you need to get them back to the ER so they can be reused, recycled, and retrograde transport is doing that as well. Let's see. So here's just a picture. It's a good break from all this flow of proteins around the cell. Here's a vesicle forming, and it turns out it's clathrin coated vesicle. You can see this is actually the clathrin ring or clathrin coat pinching out of a sphere of plasma membrane. And there's a lot of this material inside the vesicle that is cargo. It's not just random, it's specific things that interact with either the coat protein as a transmembrane protein or as a cargo receptor as a soluble protein. Here's a picture of these cage-like structures of clathrin that form in the SEM. Okay, so we're here to talk about the last step, and that is a couple of slides actually, but it's going to be how do you actually get, once you talked about how you make that vesicle, fill it with cargo, pinch it off. We've talked about all the vesicles that are going different places, but how does it actually get to the right place? How is it that the vesicle that is made by the ER always goes to the Golgi, never goes directly to the plasma membrane or to the mitochondria or someplace? So we'll start here after the vesicle is formed. It does let me add one thing. This is a clathrin-coated vesicle, and let's here look at the adaptin shown here in the green, which is conferring the specificity for cargo. Dynamin is necessary. I kind of told you for the COMP2 vesicle, it just kind of pinches off. For the clathrin vesicles, for whatever reason, you actually need a particular enzyme called dynamin to do the final pinch. Um, and so here's the vesicle that's formed. The part I wanted to focus on was this, that you get uncoating, and there is a GBP that is hydrolyzed um, uh, in the coat proteins themselves, and it leads the coat protein to come apart. It's not the same GBP that caused the assembly. It's actually hydrolysis of the coat, the clathrin coat itself. And as a result, you get what's called a naked vesicle. It's uncoated. And it's that which is able to fuse with the target organelle. So with the coat still on it, it's able to be in the cytosol, but it can't interact really with any organ. So it's not just enough for it to interact with any organelle. It has to get to the right organelle. It's thought that these vesicles travel on the cytoskeleton to move from one organelle to another. Sometimes it's a short distance. Sometimes it seems to be it's a longer distance. But here's the uncoated vesicle. There is two parts to the fusion of that with its target organelle. And both parts involve specificity. So if an organelle gets to the wrong, if a vesicle gets to the wrong target organelle, it won't fuse. And part of that is a small GTP binding protein. In the case of the clathrin-coated vesicles, it's RAB. It's another one of these GTP binding proteins. You can see they're used for multiple things. One was to assemble this, and the second is in tethering. And so the target organelle always has a tethering protein, which is kind of long, and it will bind to the RAB, and that will still not cause fusion, but it'll bring the uncoated vesicle close to the membrane. So that's tethering is the first step, and then the second step is docking. Really, I guess I don't know if I said it was two-step. I guess we can really say it's three-step. But let's talk about step two, docking. What's important there is this V snare. The V snare is covered by the coat protein, so it's never going to be able to do its job until the coat protein is removed. What its job is to form a very stable structure with another protein called T snare. The V in V snare stands for vesicle, so it's a snare protein that's on the vesicle, and the T in T snares, of course, stands for target. The T snare and the V snare have bind strongly to one another, and this is a lot of the specificity. For example, V snare from a anterograde vesicle is going to be different from a retrograde vesicle or from an endocytic vesicle. And so, or an exocytic vesicle. Each vesicle has a V snare that matches up only with its target organelle. So the target organelle, if this target is the endosome, for example, then it will have the endosome T snare and will match up with an endocytic vesicle V snare on an endocytic vesicle, and not with a vesicle somewhere else. Once you get this tight binding formed, then the lipid bilayers seem to be, just because they're close enough to each other, they just fuse. You temporarily break some interactions between lipids and form new interactions between the lipids in the vesicle and the lipid at the contact site. And so what happens to the cargo proteins? They're released from their cargo receptor. There's not any shown here, but if there are membrane cargo, the transmembrane proteins, now they're part of the target membrane. So if it's going ER to Golgi, started out as an ER transmembrane protein, ended up as a Golgi transmembrane protein. And it'll be with the same topology. So if it was inward at the ER, it's inward at the Golgi. Okay. Well, I think let's stop there. And we'll pick it up again on Monday. If you have any questions, please send them by email or you can ask them on Monday. Have a good weekend and see you Monday.